Hello, everyone. Welcome to part 2 of this series. So, in this particular section, we are going to talk about DNA sequencing and sequence alignment. So, without further ado, let's get started. So, first of all, suppose that this long stick or this long string, the colorful string, this is your DNA, right? This is your DNA, and basically you want to read it. I mean, everybody has DNA, but obviously we need to know what is inside that DNA. What is the ATCG string? What is the string with ATCG that is there in the DNA, right? So in order to read the entire DNA, entire string, what we have to do first is follow a few steps. So we have to create first many clones of your input DNA, because in order to read the DNA properly, I mean, it's a cumbersome process, it's a messy process. So it's good if we have multiple copies of your DNA and we can read as much as we want, basically. So we want a bulk of your DNA. And there is a thing called PCR. PCR means polymerase chain reaction, using which we can create many clones of our small input DNA. So after we do that, we can create random fragments of these DNA molecules using sequencing technology. So there are sequencing machines. If you pass these cloned DNAs through that machine, it will chop them up into small fragments, and basically these fragments will be pretty much random. It doesn't have any order. It's not like the first few fragments will come from the initial part and the last few fragments will come from the last part. It's nothing like that. The truth is, any fragment can come from anywhere. The first few fragments can come from the last part, and the last few fragments can also come from the first part. So there is no control right there. So there are different ways this can be done. I have named a few, and they use very different methodologies to cut up these small DNA fragments. And also, how long these fragments will be is also dependent on the technology itself. For example, in Illumina, it is around it's normally within 400 base pair, and at Oxford Nanopore, it can be over 1000 base pairs. Anyway, so after we do that, we need to store these fragments as reads in FASTQ files. So reads are nothing but strings. So basically, you cannot. You cannot really store solid fragments, right? You have to store the reads. So what is the ATCG sequence that is there in this fragment? So you will need to store those in some file, and that file is named as FASTQ file. So basically, after cloning your DNA using PCR reaction and feeding it into the sequencing machine, you will actually get a FASTQ file as output. And these files can be quite big depending on the sequencing depth. Now, sequencing depth is a concept which I will clarify after a few more slides. So bear with me. So after I stored them in the FASTQ file, these are just random reads. Now, what are you going to do? You need to be able to form the actual genome, right? You want the entire genome to be formed. You want to read your entire DNA. So the thing is, already we have the human genome available. We know how the human genome looks like. That is something we already know. Now, obviously, your genome or my genome, it will not be exactly the same as the human genome, as the original stored human genome, it will have some differences, which is not major, but they can have a lot of implications, because those differences can make us cancer prone, or they can create some autoimmune disease in us. They can create a lot of havocs, basically. So we need to be able to actually assemble our own genome. So there are different genome assembly algorithms using which we align these reads to the actual human reference genome that are stored. And there are different versions of the human reference genome. For example, AG19, AG38, and many others. Currently, I think they're using a different version. AG38, AG19 and AG38 are actually the most used ones. So yeah, those are the different versions of the reference genome. So you use some computational methods, some genome assembly algorithms in order to align these reads to the reference genome. And afterwards, there is actually a nice tool to do that as well. I forgot to mention that its name is BWA, or BWA tool. So it's widely used and available. It's a computational tool, by the way. And this kind of alignment is called comparative genome assembly, because you're comparing these reads to the reference genome. So obviously, you can now look at the consensus of the bases. So if you see that most of these bases are actually A, then this should be A. Again, if you see most of these bases are maybe G, then it should be G, and so on and so forth. So you can assemble the whole genome in that way. But there is also another class of genome assembly which is known as de novo genome assembly. Now, this is something which you will probably not use that much, because it is something used by high-level people who actually make the first first or improved version of the different species genome. So for example, suppose this is just an example. Suppose the genome of cockroach has never been assembled. So some scientists come by, they write some algorithms, and then they, create this genome from scratch. So there is no reference genome in de novo genome assembly. You only have these reads, and you somehow need to assemble those into a full-fledged human, that species genome basically. So that is done when you want to improve the current genome assembly, or that is done if for any species you don't have any genome assembly at all and you want to create one from scratch. But this is something you will probably not work with that much. At least initially, I haven't been able to work with this so far. So after you align the reads to the reference genome, you actually form a file called BAM file. So basically, you had these reads in the FASTQ file, and after aligning the FASTQ file with the reference genome, you have now successfully made a BAM file. Now, BAM file is a very nice file that has many information, which I will talk about now. So obviously, in the BAM file, you are going to have all these reads stored. And for each read, it will have many information such as the position, the sequence of the read, that means the ATCG string in the read, the mapping quality, and many other information. And if you want to handle BAM file, if you want to analyze BAM file, two famous tools are available now. One is SAMtools and one is a Python-based tool, which is PySam. So you can check them out. So there are some challenges in genome assembly. So the first challenge is sequencing error. So when you create these fragments, these are not error-free. There will be some errors, and it depends on which technology you are using. The error depends on that. And also, obviously, when you assemble the genome, when you align these reads to the reference human genome, there will be mismatch. So whether those mismatch are because of error or whether those mismatch are actual mismatches, because as I said before, your DNA and my DNA will not be completely same. So whether the mismatch is biological or whether it is a sequencing error, that is something we have to consider. Then, sometimes there will not be that many reads to work with, and that can cause some issues. So the coverage issue I will talk about again in more detail in the next slides. I have a slide for the coverage. So basically, low coverage means you don't have that many reads here. So you don't have that many consensus to work with, basically. So suppose, let me give an example. Suppose this portion, maybe this portion is covered by only one read. So only one read covers this portion. And that read is saying that, okay, this should be T. We don't actually have any way of knowing whether that T is an actual T or if it's a sequencing error. But if we have like 50 reads right here, right, 50 aligned reads in the same position, and all of them tell us that, okay, this is a T, then we are fairly confident that this is actually a T in this position. So low coverage can actually wreak havoc in these cases. Then we have the problem of repeat regions, which I will talk about in the next few slides. This is actually a big problem because in human genome, there are small regions which are repeating many times. There are a lot of repeated regions in the human genome. So it creates problems during genome assembly. I will talk about this later on in this video. So as I was saying about the BAM file, I want to give you a peek of the BAM file. So this is just one read in the BAM file. So the BAM file will store all the information. So this is the information for only one read. So if you have like 1 million read in your sample, you will have 1 million such information stored. So let me briefly explain a few of these because goal of this tutorial is not to talk about BAM file, but you need to have an idea about what's going on here. So the first thing right here, you can see right here, this is the queue name. That means the read name. Every read has such a name. And from this name, you can also infer the sequencing technology that has been used. So previously I talked about Sanger, Illumina, these things. So, with this name, you can sort of infer that this is actually Illumina style sequencing. And also there is a flag right here. It actually gives you many information such as paired end, forward strand, etc. So I will talk about the paired end thing just a little bit later on. So it sort of gives you those information about each read. Then RNA and pause are very important, these two things. So one means this read is from chromosome 1. So it came from the chromosome 1. And this is the position from where the read started. So it started in 10,005 position, the 10,005 base position of chromosome 1. And finally, there is also mapping quality. So you can see that this is 0. That means the mapping quality is actually extremely low. That means we cannot really rely on this particular read. So there are many other things here. You can go take a look. But basically, BAM file stores all this information for all the reads of your sample DNA. So now I want to talk about the problem of DNA repeat region. As I said before, that it is very problematic. Think about this line as your entire genome or DNA, right? So you can see that there is one black segment, there is one green segment, there is one gray segment, which is cool. But there are also two red segments. So assume that these red segments have exactly the same string, right? So suppose here you have AATTCC. Here you will also have AATTCC. So this is what repeat regions are. Okay, this is what repeat regions are all about. And imagine that after sequencing, so after you do sequencing like this, you have a read which looks like this. Now where are you going to align this read street to? You can align this read to this region or this region. You can do either. So how can you decide? This is a big problem because we cannot really decide in such a case. That is why people actually prefer longer read sequencing. Because if your read is long, for example, this is your read, you now have some clue, because here you can see the black portion also came, and so we know that this will surely map here, right? So that is the problem of repeat regions. So you would want longer reads ideally, but I mean longer reads have an issue. If your reads are long, then there will be more sequencing error. So it's a trade-off. It's a trade-off that you have to deal with for repeat regions. Then let's talk about paired end sequencing. As I said before, I will talk about this. So there are two types of sequencing in terms of like pairing. You will have like single end sequencing and you have paired end sequencing. So I will explain this now first. So for example, suppose this black line, right? This is one fragment. So what do I mean by one fragment? So this is one fragment. So when you cut out the DNA randomly, you get many fragments. So suppose this is one of those fragments from the DNA, right? From the genome. Now, if you are using single end sequencing, then you only probably have this read, right? This read right here, right? And which is cool. You have a read right here from this particular fragment. Now, there is an issue with this single read because, as I said before, if you have like this repeat regions and this read is not that long, right? As I said before, if you are doing longer read, you are going to have a lot of sequencing error. But if you are doing shorter read, your sequencing error will be much less, which is comforting. So if you do short reads like maybe 100 base pair or 200 base pair, then those reads, it will be sometimes hard to map because of these repeat regions. But if you do paired end sequencing, then you are going to have two reads coming from two ends of each fragment. That is basically paired end sequencing. Now this is actually much easier to deal with because you know both ends now, and you sort of can, most likely your paired end will solve the repetitive region issue because then those reads are coming from far away from each other. So you know how the starting power portion looks, you know how the ending portion looks, so it is much easier to map now. And as I said before in this slide, that fragments can be very random. Some of them can be long, some of them can be short. So you can have this kind of fragment as well, where the two reads are going to overlap, forward read and the reverse read. They will overlap. And I mean, this scenario can also occur where there is complete overlap, right? Those things can also occur. So in paired end sequencing, we know two things in the BAM file. So as I showed in the BAM file, this is the BAM file. So in the read information, for the forward read, the fragment length will be always positive. It will be a positive fragment length. And for the reverse read, it will be a negative fragment length. So for example, if this whole black fragment is maybe 400 base pair, then this fragment will have a fragment length of plus 400, and this read will have a fragment length of minus 400. That is how you sort of know that, I mean, they are from the same fragment. I mean, there is some like attributes in the BAM file which gives you the ID, the paired ID. So if you just look at this read, I mean, in that information, there will be an ID of the paired read. So this ID will also be there, so you can look that up as well. So that is paired end sequencing. Nothing, not, I would say, a big issue or a big thing, but often you will find this in the literature. So finally, let's talk about the whole genome sequencing and targeted sequencing. So first of all, let's start with whole genome sequencing. So if you sequence the entire genome, that's whole genome sequencing for you. It is not no rocket science. So what is sequencing depth or coverage? Same thing. These two are the same thing. So for example, if you look closely, each base, each point in this DNA or genome is actually marked by, on an average, I would say two fragments or two reads, right? That means the sequencing depth here is actually 2x. The sequencing depth here is 2x. If on average each base of this DNA is marked by only one read, then the coverage is 1x. So 30x is obviously pretty intuitive. So I mean, what is the advantage of like higher coverage like 30x? It is because, so let me show you the figure again. If you cover a particular base by 30 reads, then even if there is some sequencing error in some of the reads, ultimately the consensus will give you the right answer, the right base, right after the alignment. That is the advantage here. So ideally, you would want to do the deep whole genome sequencing and not the low pass whole genome sequencing. But again, there is the cost issue. So if you are doing deep whole genome sequencing or deep WGS, then you are going to have a lot of costing. The costing is very high. On the contrary, if you are doing low pass, which is less than 5x, then the cost will be much lower. So if you do 1x whole genome sequencing, then there will be, you will get approximately 10 million fragments from a single 1x sample. You can just multiply it. So for 30x, it will be 300 million fragments, basically. So the first Q file, the BAM file, everything will be 30x larger. That's it. So another type of sequencing, very famous, is called targeted sequencing. So in this case, you don't really care about the whole genome. You just care about some particular parts of the genome. So not typically, those selective regions are less than 1% of the whole genome. And because it is so less, it is such a small area, you obviously want many reads to come from those regions to be very accurate about their assembling. So typically, targeted sequencing have a coverage of like 100 to 10,000x. So it is a lot of coverage, as you can see right here. So the question is, why do people use targeted sequencing? So if you're interested in a particular region of the genome and you want to analyze mutations, changes, and other things in those regions of a particular human, it is very recommended to actually use the targeted sequencing. Because in that case, you are going to have high quality assembly and you will be able to understand what's actually going on, right? That is the thing. So that's all about this tutorial. So in the next tutorial, we are going to talk about coverage. Copy number alteration, which is basically related to coverage. The link will be given in the description. And when we talk about coverage or when we talk about copy number alteration, the first thing that comes to a bioinformatician's mind is GC bias. So what is GC bias? I will also discuss that. So see you in the next tutorial. Thank you.